since we are going to have 2-hour lecture for today. Okay, so protein is a polymer of amino acid. So when you have combination of a few amino acid, okay, then we call protein. Okay, so primary structure. Okay, so for protein we have starting primary structure, secondary structure, tertiary structure, and quaternary. Okay, so this is just normal introduction, and then so for the protein we have the one that we have discussed earlier. We have N, C, C, N, C, C, N, the repetition of this sequence. Okay, starting from N terminal to the C terminal. Okay, and then we have the carbonyl, oxygen, and amide group are trans to each other. So that means they are in the trans position, not cis trans position. The peptide bond exhibits partial double bond character. So I'm going to edit this one. Okay. Exhibits. Okay. Okay, peptide bond exhibit partial double bond character. That means we have the character. Okay, you have your proteins, your oligopeptide structure later. You have the N terminal to C terminal, and then you have the double bond CO and everything. And then when we have carboxyl group combined with the amino group, and then it can form peptide bond. So the peptide bond characteristic is almost similar to double bond character. Okay. And then okay, we have here the one that we want to discuss here. We have the torsion angle here called as pi and psi. Okay, pi is the angle between maybe Farah, you need to mute your microphone because I can hear myself from your speaker. Okay, so we have phi here, chiral carbon. Okay, chiral carbon with the amino group. Okay, with the nitrogen from amino group. So this torsion angle, we call pi. So PHI. Okay, so we have another one psi is between chiral carbon. Okay, in chiral carbon with the carbon at the carbonyl group. Okay.
Doctor?
Yes?
Can you repeat that again? It's kind of fast.
Okay. Can we have Farah mute her microphone, please? Okay. Okay, so this one, we have, maybe Anissa also need to mute your microphone. Okay. I'm going to stop the pointer because I couldn't find my pen. Okay, draw format. Okay, so here we have PHI, pi. The torsion angle between the chiral carbon, okay, this is carbon alpha, between the amino and the amino group, nitrogen and the amino group, this is called pi. And then the torsion angle, okay, the torsion angle, the torsion, T-O-R, okay, the torsion angle, okay, is the rotation, if the rotation happen between chiral carbon and the carbon at the carbonyl group here, okay, between these two, we call it psi. So to make it easier for you to remember, you can just put it here, carbon and carbon, chiral carbon. Everything is related, supposed to be attached or supposed to be related to chiral carbon, okay? So chiral carbon to carbon at the carboxyl group, we call it psi. Psi means you call it same, sama, same, okay? Same, it's carbon and carbon, okay? If you have chiral carbon, rotation or torsion angle between the chiral carbon with the nitrogen at the amino group. Okay, chiral carbon with the nitrogen at the amino group, this is pi. Okay, the torsion angle meaning that the rotation, okay, meaning that the torsion between, because once you have once you have this oligopeptide, right, arranged, right? So remember that you will have this part, okay, okay, oops. Okay, okay. So you will have this possible to have this bulky group, right? Oops. Where's the mouse? What happened? How to remove this? Where is the mouse? Maybe I should stop sharing first or what? Okay. Again, I'm going to share the screen again. Okay. So once we have this torsion angle, okay, we have this torsion angle. It's the rotation between, we have the Ahmad line, right? We have the plane there. Okay, so the torsion angle is where we want to adjust. The protein will adjust by themselves to reduce the steric hindrance. Okay, we have discussed about steric hindrance, right, last week, right? Steric hindrance where the bulky side chain they will like rotate, and then they will like crash among each other, collision between each other. We don't want this to be happen. So that's why we have the torsion angle here between the pi and psi, so that the protein itself will adjust, so that they will have the rotation at the position where it is allowed to be happen. Okay. Okay, so maybe we go for example. Okay, so we have here, in here we have carbon, right? This is the carbon alpha, and then this will be the carbon alpha. We have this will be the carbon alpha, and then this one also the carbon alpha. So what happened here, if we put the coplanar relationship, I mean the Ahmad plane, we have to put the plane there, and then what happened, if the bulky group of our group here facing similar side to this bulky group, so what happened, these two will like, okay, you are big, I'm big. So imagine we have a trailer carrying lock, okay, big lock, and then also another trailer carrying maybe what I call it, petrol. Okay, so what happened, they shouldn't be next to each other too much, because if they want to be next to each other, that means another side should go to the other side. Okay, the rotation should happen so that it won't be like scratch next to each other. So same goes to your oligopeptide later. You will try to, okay, if I have a bulky R group here, another bulky side group, R group, I will put it at the top. So that means the rotation present between this and this, it will be roughly about 180 degrees, because from here you move to the other side. If you move 90 degrees, okay, so that means it will stand up, right? So from here you move to this side, so that means you have rotated, you have performed the rotation for your R group, okay, up to 90 degrees. But if you have the rotation like from the other side, that means you have rotated 180 degrees. Okay, maybe we can see again later. Okay, let me see if I can find any example. Okay, so we go for the polypeptide and protein. Okay, if we you will have later, okay, if you combine 2 amino acid, we call it dipeptide. Okay, 3 amino acid, tripeptide, tetrapeptide, pentapeptide, hexapeptide. So for your case, to be specific, when people will ask you, the question may be asked, what is your peptide, oligopeptide name? I mean, how do we name this? So if you have 6, so that means you will mention specifically, okay, this is our hexapeptide model. If you have 7, that means you have, this is our heptapeptide model. Okay, so if you have an oligopeptide, that means you have your peptide chain longer than 12 but less than 20 amino acid residues, because above 20 amino acid residues, it can be called as mini protein. Okay, small protein. Okay, so we have here polypeptide when the chain exceeds several dozen up to 100 amino acid. Okay, proteins more than 100 amino acid. That means if you have 100 amino acid, more than 100 amino acid multiplied by 3, because one amino acid encoded by 3 codon, 3 base pair, right? Okay, 3 bases. So that means it will be 300 nucleotide. Okay, if 300 nucleotide, that means you have more than 100 amino acid. Okay, so we talk about the structure of the protein. Protein comes in monomeric protein, meaning that we have only one single type of side chain, monomer. Okay, monomeric. Okay, if we have multimeric protein, okay, it will have more than one polypeptide. Okay, so for example, insulin, later we go for insulin as an example. Insulin will have 2 polypeptide. Okay, two polypeptide. One chain and then the second chain. Okay, one is with 20 amino acid maybe. The second one with 31 amino acid. So even the size are different. Okay, the size of these two polypeptide are different. So that means this insulin is called multimeric protein because it has 2 different type of polypeptide. Okay, either it is monomultimeric or heteromultimeric. It depends on the type of polypeptide. So now for insulin, we have different sequence of 20 amino acid, and then for beta peptide, it has another different sequence of 31 amino acid. So it is called heteromultimeric. So it has different monomer, okay, different protein. Okay, we have first with alpha, second with beta chain. Okay, so these two, they are different to each other. So it is called heterodimeric because it has 2. One is monomeric protein, 2 is dimeric, 3 is trimeric. Okay, so that means the polypeptide, they have different type of polypeptide. So if you have the repetition of similar, okay, for example, alpha polypeptide, okay, this 20 amino acid, then it has another 20 amino acid, similar sequence with the above one, then we can call it as dimer. Dimer, but homodimer. Okay. Okay. So hetero is for different type of the polypeptide, different sequence of polypeptide. But if it is similar, it will be homo. Okay. What happened? Okay, we have discussed earlier about the size. How do we determine the size? What's the unit for protein size? It is called Dalton. Okay, so it will be if it is kilodalton, so the way we write it, it will be K and then capital D and then small a. Okay, if it is kilodalton. If only Dalton, then will be only D and A. Oh my God, I can't see. Where is it? What happened to this? Okay, so yeah. I will stop the annotation. Okay. So protein composition and behavior. Okay, either it is monomeric, oligomeric, or multi-subunit. So subunit is also similar to the arrangement of the protein just now. Either it is 1 subunit or 2 subunit or 3 subunit. Okay. So simple protein, peptone, peptide, albumin. Okay, conjugate protein. We have also prostatic group, non-amino acid portion, like hemoglobin. So we have a protein, and then that protein can only be function with the presence of cofactor. So in this case, the protein called globin, and then we need the heme group. Okay, this is one of the prosthetic group, so that we can make the hemoglobin being function because we have the heme group. Okay. Okay. So next one, we will have a different type of another type of protein, which is globular protein. And then we have fibrous protein. Okay. You can read all this information about globular fibrous protein as well as the previous one just now. Why? Okay. Okay, so now we have different size of protein. Insulin, one of the smallest protein with a molecular mass 5.7 kilodalton, with the number of amino acid 51 amino acid residues, and then it has two subunits that we have discussed earlier. We have one alpha polypeptide, and then we have beta polypeptide. Okay. And then they are different. And then we have cytochrome C. Okay, it's about 13 kilodalton. 13 kilodalton. The number of amino acids is much bigger than the first one, which is 104. But the number of subunit is only one. So that means it has one type of subunit. Okay, but it is 104 amino acid. It contains 104. So the number of subunit is not depending on the amino acid size, okay, numbers of amino acid, but it depends on the type of the polypeptide. If it has similar things, then it will be one type of subunit. If it has different polypeptide, that means it could be 2 or 3 or more than one subunit. Okay. So ribonuclease A from also known as bovine pancreas, the size is 13.7 kilodalton. Okay, but the difference is only 20 amino acid. So, but it contributes to 700 daltons. Okay, but it is small. I mean, only one subunit. Okay, we go for the biggest one with the lots of subunit. Glutamate dehydrogenase from bovine liver. This is 1,000 kilodalton. It's 1 million daltons. It's big. Okay, it has 8,300 amino acid residue. And then the number of subunits. So that means it has up to 40 subunits. Either it is heteromonomatimeric or homomatimeric, we don't know. We need to check on it. Okay, so.
Doctor?
Okay, yes. Amino acid. Yes.
The cytochrome C, right? Cytochrome C, it says it has one number of subunits. So it means it's a homo. I mean, it's a
the type
monomer,
of,
right? It's a monomer.
Yeah,
it's one chain.
Okay, so it doesn't mean it is monomer. Okay, we need to look at the structure. So it has only one type of polypeptide. Okay, but the maybe the arrangement. Okay, the arrangement will be different. Okay, it has similar repetition. Okay, we can check on cytochrome C structure if you want to see it. Okay, but the number subunit, the repetition of it is homo. That means it is only one, but the subunit we don't know. I mean the type, either it is homo or hetero, we don't know. Okay, we need to check on this. Okay, the repetition. I mean, you want to know the quaternary structure, right, Tanesh?
Yes.
Yeah, we need to check on it. But now we are talking about the type of subunit they have for this one. Okay, we need to check. Even for others, we need to check. But normally when we talk about this, the best way is to for us we just record like this, but we normally will go and see in the PDB data bank. Okay, Isharini asks, what does amino acid residue refer to? It is amino acid. Amino acid residue is numbers of amino acid. Okay, I'm going to share with you maybe this website. This is maybe it can help you to understand. We have here, I'm going to share the screen. You are looking at my screen, right?
Yes, doctor.
Or you are looking at my slide. Okay, can you see this PDB website? Protein
Yeah.
database?
Yes.
Okay, yeah, this is
Yes.
PDB. This is PDB where we collect all the protein data, okay, the structure data. So if you have only amino acid sequence, it will not be deposited. It cannot be deposited in this data bank. Okay, if you have the protein structure, we're talking about the structure of the protein. Okay, now if you want to know, we want to look at the cytochrome. Cytochrome. Okay, cytochrome from where? Okay, so we need to specify. Okay, maybe you want to see cytochrome from, where is it just now? Human. Okay. So we can even check and browse. Alamak. Wrong spelling. Hello? Why hang?
Hello.
What happened to my internet? PDB, please PDB. PDB. Okay, this is PDB, Protein Data Bank. Okay, now we are going to start the search. We can even put, we are supposed to put PDB ID, but if you don't know yet, then you can type. Okay, okay, cytochrome. Okay, cytochrome. The internet is so slow. I don't think we're going to have it. Okay, never mind. You can have a look at this website where you can find any of the structure that you want to see. Okay, it will, okay, the one that deposited in protein data bank, normally the structure being solved by using X-ray crystallography or cryo-EM. Okay, we will look at all those methods later. Hopefully we can finish all of this. Okay, so in detail for each of the structure, we can see from the data bank, protein data bank. Remember, we are talking about the structure. We are not talking about the sequence of amino acid. Okay, we have many sequence of amino acid, but not all the sequence. Okay, not all the protein sequence has been, not all of them have been solved. Solved means predicted. Okay, okay. We move a bit to this protein purification procedure a little bit. Okay, in protein purification is a process where we have touched earlier. Hopefully you can see my slide now. I'm showing my slide, right? Not my PDB anymore, right? You're looking at my slide?
Yes, doctor.
Okay. Yes. Now this is protein purification, where protein purification is a process where we make our protein of interest pure. We, in another way, we clean up the protein. Okay, we clean up the protein. For example, if you have, okay, your protein, okay, now you know that your mouth, you in saliva, we have amylase, right? Where the digestion of the starch happen. Okay, so amylase is the basic enzyme, is the common enzyme that you have heard before. And amylase in our mouth. Okay, in our mouth we have in saliva, and then inside saliva we have amylase. So it's not only there must be a few other things present in the saliva, right? So the thing that we want to use to purify, it means we want to separate amylase then the other things. For example, you spit your a container of the what is it? I don't know. When I use the pointer, I lost my mouse. So maybe I should close the pointer or what? Okay. Wait. You may pause for. Okay. So if we have this, okay, we have this beaker. Okay, inside this beaker, we may have like impurities. Okay, the one that we want is blue color. Okay, this will be the amylase. Okay, so the one impurities may be the black color, and the red color, and then combination with the yellow color, and then the black color. Okay, maybe all these black, but remember the amylase, okay, the amylase that the targeted amylase that we want just now is in this color, okay, amylase. Okay, so how do we make it pure? Is by having a process called purification. Okay, so this process called purification is a process to separate the protein, okay, the target protein from the impurities. Okay, the impurities here, we call it as crude. This is crude enzyme. Okay. Crude protein or crude enzyme. Okay. So when we say crude, that means it is from the original source or from the one which is still not pure. Okay, so what happened here, when we try to use this, one of the technique called here is chromatography technique. It is called chromatography. Chromatography technique. Okay, so what happened in this chromatography technique? This yellow color here represent the matrix, the resin here. The resin, or also called the matrix. Okay. So here, all the impurities, all the protein will be injected in this chromatography column. Okay, it is a glass column. Okay, what happened when we put all of this inside this? Okay, this column with the help of this resin, it will separate the protein according to the type of column that we use, type of resin that we use. Okay, so if we use chromatography, where the chromatography, the type of resin here, will separate protein according to the size, then protein will be separated based on the size. Okay, if we use the type of resin where it will separate the protein according to the charge, then protein will be separated according to the charge. So what happened at the bottom here, we will have a fraction collector. This part called fraction collector. Fraction collector here, we will put the test tube. We put the tube where the tube one, initially when we have the first step, we call equilibration. And then second step, we have sample application. Okay, when we apply the sample. Okay, the third step will be washing step. Okay, last step will be, maybe the second last step will be elution step. Final step will be regeneration step. Okay, where we use regeneration because we want to reuse the column. Okay, so when we say equilibration, that means we prepare the column, we prepare the resin here, so that the column here will ready to accept the sample. Okay, so for now, if we want to separate all these impurities just now, okay, we separate the impurities just now here. So if we want to separate the protein according to the size, so that means we have used gel filtration technique. Okay, so that means the protein with bigger size will come out first. Okay, the principle for each chromatography will be different. We can separate according to the size, charge, and also the affinity. Okay, affinity. So I will share with you the video, a short video to differentiate between each of these. And then we have also depending on the hydrophobicity technique. Ion, it is based on the hydrophobicity, and then this is based on the size, this one based on the charge, this one based on the affinity. Okay, so what happened here, we can see here the fraction collector will collect the protein. Okay, the first two tubes, or maybe the earlier part will be the equilibration buffer will come out first. Okay, because we have put this metric with the equilibration buffer. And then the second part, when we start to apply our sample, okay, this green protein will come out first. So instead of having a mixture like this, the protein come out according to the time that we have determined, we have put earlier. Okay, if it is according to the size, that means the bigger protein will come out first. And then second, for tube number 5 and 6, this brown color protein we get into, and then we will have another fraction, another tubes here for the protein, black protein. So that means we need to check in which fraction, we need to check in which tubes will contain our amylase, the blue color just now. Okay, my question is amylase an enzyme or not? Is amylase an enzyme or not?
Yes.
Yes.
Yes,
Can
doctor.
it be detected using specific assay? Can we measure the presence of amylase? Do we have assay system for amylase to detect the presence of amylase? How do we detect the presence of course, each of the tube will be colorless, right? Everything will be colorless. We can't see what the difference between each tube. But we can know, we will know the presence of amylase in which tube by performing the assay. Okay, we call it amylase assay. So what will be the substrate when we do the amylase assay? Substrate for amylase. Okay.
Starch.
So that means if we put starch in tube 3 and 4, if there is no reaction, that means 3 and 4 is not amylase, right? And then we can change to tube number 5 and 6. If there is a reaction with the starch, that means it's possible to have amylase. Okay, it is an amylase. So we go to tube number 7, 8, until we are going to have maybe 100 tubes, you see, to get to know all this step. Okay, so maybe I will stop for this one. I will show you the video for a while. Hopefully you can okay. Oops. Stop sharing this one. Share only the Safari. Okay. We go for gel filtration. Gel filtration chromatography. Okay. Maybe YouTube is just a short one. Okay. Principles. I want to find the short one. Size exclusion. So another name for gel filtration will be size exclusion chromatography. Where is the... It's supposed to be GE. Okay, this one. Okay. Size exclusion chromatography. What happened to the internet. Okay. So this one.
Separation in affinity chromatography depends upon the reversible adsorption of biomolecules through biospecific interactions on the ligand.
What happened?
Most affinity chromatography experiments are performed in three main stages: equilibration, sample application and wash, and dilution. The first step is the equilibration of the stationary phase to the desired start conditions. The second step is sample application and wash. The goal in this step is to bind the target molecules and wash out all
Okay, this is not affinity. This is affinity chromatography. I want to find the size exclusion chromatography. Why it doesn't work well? YouTube. Exclusion chromatography. Okay, this one.
One of the great challenges in this world is knowing enough about a subject to
Okay.
think you're right.
So now, okay, this is gel filtration. So we separate the protein according to
separation
the
in
size.
gel filtration chromatography is based on the differences in sizes from biomolecules as they pass through a column packed with a chromatographic medium, which is a gel. Molecules larger than the pores in the gel are unable to diffuse into the gel and are confined to the solution surrounding the beads. Molecules which range in size between the very big, excluded from the gel, and the very small can penetrate the pores to varying degrees based on their size. Access to the pores is limited by steric hindrance. If a molecule is smaller than the smallest of the pores in the gel, it will be able to enter the total pore volume.
Okay, so you may not be able to catch up, but I will explain. Okay, this will be the example of the glass column just now that we have in the slide. Okay, so the glass column inside, if you can see there is a white color matrix here. This is called matrix or gel inside. Okay, the matrix or resin here. Okay, so what happened here? Inside that matrix, we have these pores. Okay, the resin just now, inside it has the pores. It has the pore size. Okay, so depending on the size, and then depending on the type of protein, the size of protein, meaning that the molecular weight, the mass of protein, the Dalton size. Okay, I repeat, depending on the Dalton size of your protein, the molecular weight of the protein, you will select the proper pores of the matrix. Okay, so for example here, we have a mixture of protein in the crude protein. The crude protein, of course, it has many types of for this case, it has red color, orange color, and yellow color protein, right? A mixture of protein. So what happened in gel filtration principle, okay, the small protein will go out later, okay? The big protein will come out earlier. So if you look at this picture, you see, this is small size of the pores. So that means only the small protein can get into these pores. So the big protein, it cannot get into this. That's why it moves faster than the small protein. Okay, the small protein here, it has time. It will have time to get into the pores. Okay, so for the big protein, it cannot fit into this pores, into these small pores here. That's why it moves faster. Okay, so for this case, that's why, okay, when you look at the movement of the protein, okay, the yellow color protein is the smallest protein, will take time to get out from the pores. Okay, but the orange protein has time, okay, to get into the second pores. I mean the second large pores. Okay, so but the big protein now, even this pore it cannot fix. This pores it cannot fix. This pores it cannot fix. That's why it moves faster. Okay. So for this case, okay, we can see here, okay, the movement of the protein. The red protein definitely will move faster, and then it will go to the collection tube. It will go to the test tube earlier than the other protein. So that's why from this chromatograph, okay, chromatogram, we can see the red protein. Red protein can be detected earlier. You see, the red protein, these absorbance here represent the protein absorbance. Okay, once the red protein come out, you can see, you can detect the presence of the protein by having the absorbance at 280 nanometer. Okay, so now that's why we have the peak. Peak here represent the protein, the biggest protein. That's why we have the peak earlier. Okay, and then after that we can see definitely the orange protein will come out as the second size of protein, right? Second size of protein, because it will move faster than the smallest protein. So that's why we can see here, okay, the orange peak has been detected here. Okay, the second biggest protein, the intermediate size of protein. Okay, finally, since the smallest protein take time to get into the pores, okay, jalan-jalan into the pores here, I mean, sightseeing inside the pores, that's why it come up later. Okay, so for this case, we can see once the yellow protein come out, it can be detected using chromatogram. So for this case, if we are separating the protein, I mean the amylase just now, we need to check either this peak, or this yellow peak is the amylase. So that's why before we start doing the purification, we need to know the size of the target protein. Remember, we need to know the size of the target protein. For our case, we want to know, we have the amylase just now as our target protein. That means we need to know the size of the amylase. Check on the molecular weight of amylase, so that we know where to expect to find amylase. Okay, so if you know the size of amylase, then you know to use the correct metric with the correct pores, with the correct pore size. Okay, if you don't know, if we know that amylase is not the biggest protein, may not be the biggest protein. Okay, wait. Okay.
Yatimi Hakim.
Okay, so again, sorry, many distraction. Where is it? Okay, so we have the size of protein. I'm sharing the screen, right? The whole screen, right? Okay, so now you have amylase just now, and then your amylase, you know the substrate for amylase will be starch. Then you can start to quantify or detect your amylase. Maybe the tips here for orange, for yellow, for red protein, you can detect. Is there any reaction with amylase? No. So that means the first peak is not your amylase. Go to the second peak. Is this your amylase? No. Then not the orange peak. Go for the yellow one. Is there any amylase activity? Yes. So that means the earlier part now has been purified. What you want to purify now, you want to focus now is the yellow proteins. Okay? Because now, okay, if you look at your slide earlier here, we know that, okay, once I check on the protein activity, I mean the enzyme activity, the present of the amylase activity only present when we assay the black color protein. I mean the later fraction, not the earlier fraction. So that means earlier you have a lot of impurities, right? Earlier you have a lot of impurities with all these amylase, right? In the beaker here, you have a lot of other things. Now at least you have separated these two, okay, in separate tube, and then what's left? Maybe another impurities, okay, because the black color already being purified, okay, the red color also being removed. Okay, so this is a little bit on the purification. At least you get to know what is purification means here. Is it okay for the purification part, guys?
Yes.
Yes, doctor.
Yes, doctor.
Okay, so now we move for the next slide.
Sir, I want to ask, didn't catch up for substance in test tube 1 and 2. What substance?
Okay, so remember that we have equilibration tubes earlier, equilibration step. So initially the column being packed with the buffer, okay, the resin cannot be dried up. It should be in the buffer. So this, initially the resin here, the pores here inside, need to be packed with the buffer. So now the first two tubes, because since we are injecting our sample here, so what happened? The buffer, initial buffer in this will be coming out earlier, right? Because once we put this is not, do not imagine this like a pipe, you know, like a water flow from the pipe. This will take really slow process. Okay, we need to control the flow rate here, flow rate. Okay, some of the flow rate, it can go up to 0.5 mil per minute. Okay, so once we have put the sample, when we have applied the sample, okay, all the buffer, initial buffer in this tube will go up earlier. Okay, once we reach tube number 4, number 5, number whatever number, up to maybe 100 tubes here, maybe more than 100 tubes here, then you can see we can detect the presence of the protein in this individual tube. Okay, who's asking just now?
All right.
Okay. Okay, so now don't get confused with the two tubes here, because we cannot leave the resin like that. It's supposed to be in a buffer solution. Otherwise, it will not be, some of the resin, it need to be charged. Remember that we want if we learn about ion exchange chromatography a little bit last week, we want our resin to be negatively charged or positively charged. So they are basically in the buffer. They are not just something dry. Okay. Okay, so how do we move this? Oops. Okay, so we move on for the next slide. Okay, once we know we have separated the protein according to the size. Okay, we have separated protein according to the size. So we have this gel system. Okay, we have this gel system. Okay, this is gel. Okay, to detect the presence of the protein, we use native gel. We have paste gel. Either it is native paste or SDS paste. Native paste or SDS paste. Okay, so for paste basically represent polyacrylamide gel electrophoresis. Okay, it is polyacrylamide gel, polyacrylamide gel electrophoresis. Oh my God, why I cannot type? Gel electrophoresis. Okay, I put it in the chat. Okay, so this is gel, polyacrylamide gel electrophoresis. Okay, if it is SDS, that means we have sodium didosyl sulfate polyacrylamide gel electrophoresis. This native paste and SDS paste, they are similar gel, except the composition of the gel. Okay, except the composition of the gel will be different. Okay, so gel here is a really thin, the gel is really thin, and then we have the cast to make the gel. The thickness of the gel is only 1 mm, 1 millimeter, or 0.75 millimeter. Okay, so it's small, it's a really thin and then colorless. Okay, so what happened? This gel will be used to detect the size of the protein, or the protein size. We can detect the size of the protein using molecular weight here. And then the biggest size here will be 1,110 kilodalton, okay, or 1,100 110,000 kilodalton, and then 90,000 dalton, 70,000 dalton. Yeah, so this is the thing. So normally we didn't label like this, we call it 90 kilodalton, 70 kilodalton, 50 kilodalton. We didn't write the 000. Okay. So for this case, you see the charge move from the negative charge to positive. So that when we load our gel, when we load up the gel with the protein, first we load the protein. What happened here? Why I cannot change the color? Okay, so we load our protein here first. Okay, let's say we have purified the protein just now. We have separated the protein according to the size just now, right? Okay, so now what happened? Maybe we want to try tube number 2, 3, 4, ataupun 4, 5 until whatever size that we have detected. Then we load into this gel. So what happened? We are going to apply the current from negative to positive. The gel will move. The gel will move from here, from the top here to the bottom. So for this case, the bigger protein, okay, big protein move slower. Okay, the biggest protein size, okay, move slowest. Okay, at the slowest time. Okay. Okay, so that means the biggest protein will move slow at the slowest time than the others. So if you have a small protein, the small protein will move faster. Okay, so because inside this acrylamide, okay, there are also a pores here. There will be also the pores in polyacrylamide gel system. So that means the gel will move according to the charge and according to the size. So for this case, the biggest protein will move slowly. That's why we have the marker here. This we call marker. Okay, the ladder. We call it protein marker. Like a ruler, protein ruler. Without this ruler, you don't know the size. Okay, if your amylase just now, the size is 45 kilodalton. So that means you should expect the amylase will give you, or will give the band between this position, right? Because this is between 50 and 30 kilodalton. 45 should be in between this. Okay, if your protein size, okay, for example, this one, what will be the size of this protein? Because it has been detected at this position, what will be the molecular weight of this protein? Are you
90 kilodalton.
Yes, it will be 90 kilodalton because the band here, the protein band here, stop at this position. And then this position is corresponding to the 90 kilodalton. So you should be able to differentiate the size because we have the ladder here, we have the marker here, we have the ruler here. Without the ruler, we don't know. So it is important for us to know the size of the protein before start doing protein purification. That it makes sense, right? Why do you need to know you need to predict the protein size, the peptide size of yours, so that you do not purify blindly. If maybe I ask you, what will be the size of your target protein? I don't know. So what are you purifying? What will be the size? How do you detect if you have multiple bands here? You may have the bands here, you may have the bands here, somewhere around here, somewhere around here. And then without knowing the expected band size, how do you differentiate? Because all of them will give you same protein bands. I mean, the color will be the same. Okay, I'm going to show you okay, hopefully you still looking at my screen, right?
Yes.
Yes, doctor.
Okay, so this will be the gel. You see, how do you know your protein gel? So without knowing okay, you have this, the size is not only with 110 kilodalton, okay, but it comes in different sizes depending on the marker that you use. Okay, so now, how do you know this is what size, this is what size, when will be your protein?
Doctor, we are seeing your PowerPoint.
Okay, you are not seeing my internet. Okay, I'm stop sharing this. Okay, this one. Can you see?
Yes, doctor.
Yes, doctor.
Okay, so now you have this one with, it comes with different molecular weight ruler size. Okay, definitely normally we M represent the marker, the ruler. Okay, so now you can see these are the bands, the protein bands. If you do not know the target protein, how do you know your protein has been detected or not, present or not? Okay, so the size of the protein is important to check whether your protein has been detected or not. Even in terms of the purities of the protein, if you look at in this band, right? Okay, so in this case, you see, this is the crude, the band size for the crude. Your target protein is 97 kilodalton, for example. Then you can see clearly all these 97 kilodalton present in here. Okay, thick band. Okay, but once you have conducted the purification, it will make it purer than before. So it start to be pure. Okay, this one, this is the crude. Okay, this one maybe less band become what we call it being detected, because it become purer. And then this one, the fourth lane, I mean the fourth well, okay, the fourth well, okay, we can see the other impurities also being removed. Okay, when we look at the fifth well, okay, the fifth well, it looks much better because the other impurities has been the other impurities have been removed, right? Yeah, the only target protein here will be much better than the other impurities. You can even polish this to make it only the target protein present, and then the rest of other things will be removed. So yeah, this is the thing. How do we purify the protein? You can see the difference between the impurities with the one purified. Okay, what the difference?
Doctor, I have a question here.
Okay.
So once we add our protein into the column number 2, will we take that will we take what we got from number 2 and add that into number 3 to make it purify like that, or how is it actually, doctor?
Okay, the process will be, okay, for example, this is how do we check the quality of your purification. Okay, you have your crude protein initially, this will be the crude protein. Okay, you have conducted the purification strategy, and then all of the tubes you will run in this one gel. It is not first run and then you wait, and then after finish purification you run the second well. No, it is one shot experiment. So that means you have your crude here, and then once you want to monitor the quality of purification, you run one shot. You run the tube number, this is the first one, the crude, right? And then the second, and the third and fourth lane here, maybe the one that you use for column, whatever column. I mean the fraction collector just now. In fraction collector, you can see the column, right? Yeah, so every time you introduce a new technique, or you want to check the quality of your purification, and then you will use gel electrophoresis here to check either you are getting other impurities, are you observing other impurities or not? Okay, so am I answering you, Deva?
Yes, doctor.
Yeah, so it is one gel, it's one time, because the current need to, your protein need to be loaded. Okay, if you are looking here, we are stacking gel and running gel, right? So you're going to load the first well, the first well with the marker, and then here will be the second well with your crude protein, this one with the protein with the washing step ataupun flow through, this one with the washing step, this one with the elution step, something like that. So normally the pure, the most pure here will be the one in the elution step. Okay, we can see a lot of protein bands because the aim here, I want to show you the protein bands. Okay, we have different kind of protein band. So this is really important for you before you start doing purification. You need to know the protein size. What will be the size? Farah said, what difference between column 1 and 2? Which column, Farah? Again, Farah? Okay, maybe you want to speak up with which one? Are you referring to the slide or you are referring to the gel that we are showing in the website? Are you referring to this slide? Okay, so now you get the answer, right? So slide one is for the marker M. Okay, this one is for the protein marker. Okay, this will be the marker. This one will be the sample. Okay, so if you have the crude, you can load the crude because it is not only 2 wells. It will be 1 to 10 wells. Okay, maybe 11 or 12 wells, depending on the comb. Okay, so now if we go back to the okay, so if we go back to the slide about the protein molecular mass, okay, you can see that glutamate dehydrogenase will be at the top of the gel, SDS page, right? SDS gel or page gel, because this will be the biggest size. Okay, and then the second one will be this one, these two, RNA polymerase and ferritin. So it's hard to differentiate if they are all run together. Okay, 1 million Dalton is okay because it will be at the top. Okay, but for ferritin and RNA polymerase, it will be hard because these two proteins, they are similar in size. They will stop exactly overlapping at the 450 kilodalton marker. Okay, then we have serine albumin at 68 kilodalton, immunoglobulin G at 145. This will be very distinct size. Okay, so yeah, this will be the size of the protein. Okay, what else? Okay, this one done. Okay, so different type of protein come in different function. For globular type of protein, fibrous protein, for hemoglobin, for transport, oxygen transport, myoglobin, for storage of oxygen. This all you can read because it's just a note. Okay, so this one about the size of the protein, we have also discussed vitamin synthesis with 600 kilodalton here. And then the subunit organization, you can see alpha beta, insulin alpha beta, because it has 2 subunits. Okay, cytochrome C just now, one type of subunit. Okay, even though the size is 12.5 kilodalton. Okay. Okay, so this will be the size according, if we look at the protein using the tertiary structure, okay, quaternary structure. You can see the insulin here, cytochrome C, ribonuclease. Okay, so maybe all of this come with the cofactor or prosthetic group to make it work. You can see the heme here everywhere. Glutamine synthesis definitely is the biggest one, because glutamine synthesis just now we 600 kilodalton, and then you can see the quaternary structure looks really big, and then it has 12 similar subunit. Okay, alpha subunit, but it has 12 copies. Okay, 12 copies to make it this. Okay. And then, okay, protein cluster, fibrous protein, globular protein. also membrane protein. Okay. Yeah, you can see here fibrous protein, myoglobin protein, which is globular protein for oxygen transport, and then we can see the membrane protein also. We can find the membrane protein at the phospholipid bilayer here. Okay, we have many types of protein function. Okay, normally we learn about enzyme. We will have enzyme topic later, like amylase. We have used amylase as example just now. And then we have structural protein. Okay, protein act as a structural protein like collagen and keratin. They play this type of protein play roles to make up structure. Okay, we have antibodies as immune protein, transport and storage protein like lipoprotein or albumin, glutathione and receptor protein like G protein, muscle contraction, actin, myosin, dennin. Okay. Okay. Yeah, this one also you can read. I'm not going to read the notes. Okay, protein structure. We have discussed also protein structure, primary protein structure, sequence of amino acid residue. Secondary structure, we are going to discuss about alpha helix and beta sheet in the next lecture. And then we have tertiary structure, where the secondary structure interact and pack into a globular subunit. And then we have quaternary structure, where we have the association between two or more polypeptide chain to form multi-subunit protein molecule. Okay, so for the protein to be active, okay, for protein to be active, it has to be in tertiary and quaternary structure. When it is in the secondary structure or primary structure, the protein, okay, they are not active. Okay, it cannot be functioning in the primary and secondary structure. They can only work when it is in the tertiary or quaternary structure. Okay. So yeah, do you want to take a break, guys? For a while, we can pause for a while.
Yes.
Yes. Okay. Maybe Arif can pause for a while the record, and then I can answer any question. For those who want to go to the bathroom for a while, you may go. Yeah, Arif, are you still recording? Okay. Okay. Any question so far? We still have plenty of topics basically to be covered. Hopefully, we don't have to come out with the extra class. Hopefully, because we haven't touched at all the carbohydrate, even the protein and enzyme. We haven't finished it yet. So you can type any question, guys. Arif ni hilang ke mana So stop recording sekejap. So we have the protein starting from N-terminus to C-terminus, and then we can see numbers of interaction can be present in the structure. So for this case, we may have
Doctor?
Yes.
I'm sorry, but I'm not seeing the slides.
Oh, really? What am I sharing?
How the video?
Oh, no, I'm showing the slide. Okay. Are you showing the slide now?
Yes.
Okay. Yeah, we have here N-terminus to C-terminus. So normally N-terminus with the blue color. Yeah, and then carbon at the C-terminus we put it with the red color here. So what happened? We can see some of the for this case, for the short one, we don't maybe we can consider they put all the glycine here. Okay, so there will be here no side chain, but you will need to attach your side chain depending on the amino acid that you have selected. Okay, same polypeptide chain with alpha helix represented as a coil purple ribbon. Okay, so for this case, we had a secondary structure represented by alpha helix or beta sheet. So you can make a turn. It can present. It can be presented. Okay. as the alpha helix shape. But with the 7 or 6 selection of amino acid that you have got, I don't think you can form all of this, even though it's possible to have the one turn, okay, because one turn will have around 3.6, roughly 4 amino acid to make one turn, okay. But don't stress too much. Okay, we need to have cone shape. We need to have something like beta shape. No, we don't have to do that. Okay. So this is the secondary structure for beta sheet conformation. You can see here we have the, for this case, we have the N-terminus to C-terminus here. Okay, it is not always, we need to start. It is a must to start from left to the right. No, it depends on the amino group, that the first amino group. Okay, so that will be the N-terminal, and then the one at the end will be at the C-terminal. Okay, so we can see here 3 polypeptide chain forming beta sheet structure. You see we have the first polypeptide, second polypeptide, and the third one, and then they are all connected to form beta sheet using hydrogen bond. Okay, we can see here hydrogen bond formation. Okay, so here it is parallel or anti-parallel when we look at this. This is unlabeled, but what do you think about the beta sheet here? It is parallel or anti-parallel arrangement?
Anti-parallel.
Anti-parallel.
Yeah, you can see it, right? Definitely this one because N-terminal, C-terminal, and then N-terminal again. Definitely this will be anti-parallel, okay? Okay, okay, for the primary structure, it is all the peptide bond. You can see the oligopeptide model that you'll be constructing later will be in the primary structure. Okay, and then you have secondary structure, beta pleated sheet, and then alpha helix. So don't aim too much to make a secondary structure like alpha helix or beta sheet or even loop. Okay, and then the tertiary structure here, you can see all the combination between the beta pleated sheet and the alpha helix and also the loop here. Okay, another thing to have in mind, okay, you will need to know what kind of amino acid suitable to be at the loop position. You know, loop is not a beta sheet, and then loop is not an alpha helix. So loop is just like a connection between maybe the first area, I mean the first domain and second domains or something. It's just like it present there to make a turn and everything. Okay, so for this case, what will be the most suitable amino acid to be present in the loop? You have 20 common amino acid. What will be the most suitable? I mean, your suggestion. What kind of amino acid to be at the loop position? Any suggestion?
16.
16. Why?
Because we can form disulfide bridge.
Okay. But not really correct one. Okay. So the keyword here, the loop should be really flexible, because the loop should be able to maintain the structure. Either, okay, we go this side, we go to the other side, you know, all the torsion angle between this subunit and the other subunit and everything. The loop normally should be really flexible enough. So what will be the among the 20 common amino acid? What will be which amino acid will be the most suitable candidate, okay, to form loop or to make turn? I mean to make a U-turn. Okay. Do you understand the question, or you still got lost with the question?
Glycine, doctor.
Glycine, why?
It's the simplest amino acid.
Okay, yeah. So glycine, alanine, all these small amino acid. I mean, why we call it small? Because it has, they have the R group, small R group, right? So imagine if you have like Histidine. Wow, a super long trailer there. How can, do you think in the U-turn or in the traffic light and everywhere, do you think a trailer easy to make a U-turn? No. So that's why we need something small and really flexible to make a turn. So here for this case, you can suggest all these small and less bulky amino acid. This will be the most suitable position where the turn can happen. Okay, so imagine motorcycle, okay, maybe represented by glycine versus a lorry, a big lorry, okay, or with a trailer or something. Okay, so all of this, bicycle versus trailer, which one will be easier to make a turn or to make a U-turn in the traffic light? Definitely something small like motorbike, right? Or normal car, a simple car. So that's why we need to think about the R group there. We are looking for the R group which is less bulky. I mean, I would say small amino acid, small protein, small amino acid with a small R group. Then it can easily make a turn. Okay. So even in your protein, in your oligopeptide structure, you can even suggest, okay, this amino acid will be, this oligopeptide may be flexible due to the presence of this alanine. If we want to make any turn or any loop here, this will be the position where it is suitable. Okay. Then we have this one, hemoglobin. Hemoglobin with the ribbon. Okay, in the protein structure, we can see the protein can be represented by ball and stick. Okay, but the one that here is based on the ball and stick, with the ball and the stick to represent the protein structure. And then it can also be represented by wire. This will be by using wire. Still, they are similar things, but this one represented by wire. And then here we use ribbon style, cartoon and ribbon. So that means the amino acid, the protein structure is represented by using ribbon style. For this case, for hemoglobin, we have 4 types of subunit here: polypeptide 1, 2, chain 3, and then chain 4. Okay. Okay, here we have the 1 turn here. We have discussed about 1 turn. 1 turn of helix is about 0.54 nanometer per turn, which has 3.6 amino acid unit per turn. So that means it requires at least 4 amino acid to make a turn. 1 turn represents from here to here. Okay. And then we have here N-terminal to the C-terminal and CCN, CCN. Remember when we have when we have like this, so easy to show. Anti-parallel, and then N-terminal to the C-terminal. But once we have like this, you should be able to show which one is the N-terminal, which one will be the C-terminal. Okay, for this case, for B, which one will be the C-terminal to the N-terminal? From the top to bottom or from bottom to the top?
Bottom.
It is from the bottom to the top, right? Because we have the N-terminal to the C-terminal at the end here. Okay, so this one, when we have the arrangement of the parallel, okay, N-terminal arranged, they are arranged in the same direction, in the same position. Then we have the hydrogen bond formation is slightly, it's not in the parallel side. It will be a little bit of not straight anymore. Okay, and then here we have beta pleated sheet from N-terminal to C-terminal arrangement. Okay. Okay, so we are at 3:30 now. We should stop because we are almost half of the way here. Yeah, we still have few more to go for this slide, but this will be among the longest slide, I mean information from this topic. But remember, even though you are doing your oligopeptide model, the topic will be covered starting from classification, starting from the oligopeptide or peptide bond formation until now, and even until next topic. Okay, so yeah, regarding the torsion angle, maybe we want to discuss a bit. Okay, so even for here, right? So we have this complex tertiary structure here. So they are all maintained because of the torsion angle and then the interaction present in the protein. Okay, for example, in this complex structure, it involves what I call it, hydrogen bond formation, van der Waals, hydrophobic, and everything. So apart then all these weak molecular forces, they are also controlled by the torsion angle. Okay, so maybe you want to have a look and explore yourself about the Rama. Okay, so this will be the so this will be the Ramachandran plot when we talk about the torsion angle just now. Okay, so Ramachandran plot here. So it talk about the torsion angle, either that arrangement, that torsion, psi and pi torsion is within the allowed region or disallowed region. So maybe this one. Okay, so if it is more than 80%, more than 90%, that means it is acceptable. I mean the torsion present, okay, they will be here, will be the most favored region. Okay, if it is more than 90%, that means the structure or the torsion present is acceptable. Okay, residue addition allowed region, okay, 4.5%. So if it is more than 80%, 90% here, that means the predicted structure can be accepted. Okay, you may have a look at this Ramachandran plot, okay, to verify the structure. Even though the structure can be, the torsion can be present, you see the pi degree, psi degree, I mean the rotation at the axis, at the, I'm applying, right? Okay. Yeah, the good structure, the bad structure, everything. And then maybe this one, the one that we want to check, right? Again, maybe basic search again, if you can get it. Yeah, we go for lysozyme. Okay, so this will be the lysozyme protein. Okay, so lysozyme in the PDB, that means this lysozyme, it has this structure, and then it has been solved using X-ray diffraction method with the quality of the resolution 1.77 angstrom. Okay, so this is trioiodide derivative from hen egg white lysozyme. Okay, it has been published in 2002 in Acta Crystallography, D Biology Crystallography. Okay, so human lysozyme with the glutamate acid, glutamate acid, what will be the code for glutamate acid last time? It's supposed to be E, right?
Yeah.
E,
It is
R.
E.
E, right? Okay.
Yes.
So E35A, because this alanine, right? Yeah, so at least when you look at this, you do understand. Oh, okay. Now you know how to read this. Okay, even you can read the resolution here. The smaller the number of angstrom here, that means the better the structure. I mean the structure is acceptable, and then it's well elucidated. It is well elucidated. Okay, it is not a structure prediction. It is elucidated structure. It is a confirmed structure. So when we learn about the protein structure, at least we know how does the protein work. We know how the protein works. Oh, okay, this is how it works. Because this amino acid responsible to do what? This amino acid represent to do what? Like amylase just now, right? You know the amylase can hydrolyze starch. but how? Okay, so that's why when we know about the protein structure study, then we will understand, okay, that amino acid that histidine at the position 200 interact with glycine at position 180, they can form what, and then they make it like an active site so that the enzyme can be functioning. Okay, but for us, if we want to have a good structure, we need to use X-ray diffraction technique, and then also the quality that can be accepted by PDB, protein data bank, it should be within less than 5. Okay, the higher the number, that means the resolution is not good. I mean the elucidation is not good enough. Okay, these are all really good protein. 1.8, they are all using X-ray diffraction. We can even predict the structure. We don't even have to use X-ray, because when we need to use X-ray, that means we need to have a crystal structure. So we will cover that part sooner or later in the next lecture. But at least I want to show you, this is the protein data bank, where the protein data bank will keep the structure of the protein. What you want to know? Maybe, okay, insulin. Insulin from where? So maybe just generate insulin, then the structure of insulin will come out. Okay. So crystallography of insulin, energy generation. Okay, 1.6 Armstrong. Homo sapien insulin. This one, Bombyx mori. And then it used NMR. It is not X-ray. So there are different type of, this one is nuclear magnetic resonance technique to solve the structure. Okay. So we have also protein homology modeling to predict the structure. And then we have cryo-EM technique to predict the structure. But normally, the one accepted is only from X-ray crystallography. Okay, that's it, guys. Yeah, you can, Arif, you can stop recording now. So we have any question? For our test one, we only need to be tested for protein and carbohydrate, not amino acid, right? No, it's together. Amino acid, protein, carbohydrate. Okay, carbohydrate will be under monosaccharide, disaccharide, polysaccharide, and then different type of. We will plan maybe on week 5 and 6. We will cover 2 weeks for carbohydrate. So that means we need to finish this by this week, or maybe next week, 1 more week. Okay. So yeah, if look at the slide, you have only that kind of slide. But when we discuss, we have many things to be discussed, and then many websites have been opened, so that you get to know everything about protein world. Okay, because this is about the protein structure, and then another website you can use Gene Bank. Okay, NCBI Gene Bank. It is by the National Center for Biotechnology Information. So this will be, if you want to know the protein structure, the genome and the protein. Okay, you can even type here, maybe insulin just now. Yeah. So here it will only give you the sequence, but may not be shared with you the protein structure. Okay, like here we have more than the one in the protein structure database just now. So this will only share with you the protein sequence. Okay. Okay, insulin from hamster, ovis aries, insulin from myotis brandy, from common squirrel, monkey, ostrich, elephant. Okay. And then we have also from Uniprot database. So this is also a curated database. So we can even type whatever protein that we want to know, and then this one with the referred, with the reference, with the referred, we can refer to. Okay, for this case, if you find any protein, unique protein you want to share, You can even deposit your protein. Okay, you can deposit your protein here. But if you plan to deposit the protein from human, amylase from human, no need, because there are definitely plenty of the information about the amylase in human being deposited in here. Unless you want to do mutation with that kind of amylase, then it was to be deposited in here so that you can secure that kind of information under your name. Okay. Yeah. Arif, have you stopped recording so that we